introduction. So hello everyone, I'm one of the co-organizers of the Illuminating Druggable Genome DREAM Drug-Kinase Prediction Challenge, and today I would like to give you the challenge overview as well as summarize the results of the recently completed first round. A little bit about the challenge. So it was selected by the program committee as well as the community among tens of applications in the summer last year. So it is jointly organized by DREAM and Sage Bionetworks, as well as the Institute for Molecular Medicine Finland, Illuminating Druggable Genome Consortium, Structural Genomics Consortium, UNC-Schellman School of Pharmacy, Icahn School of Medicine, and the BRIC Research Center at the University of Copenhagen. So you can see that we have quite many institutions involved. And we also have a scientific advisory board consisting of 4 experts in the field: Christer Wenneberg, Avner Schlesinger, David Drury, and Tudor Oprea. So our challenge is focused on the prediction of binding affinities between drug compounds and proteins. And this is because understanding the mode of action of a chemical compound, including its potential off-targets, is really a critical part of the drug discovery efforts. And as we all know, it is quite important for the design of effective and safe therapies. However, experimental drug bioactivity profiling is still relatively time-consuming and expensive, even if we use modern high-throughput technologies. And furthermore, the size of the chemical universe is really enormous, so we also simply cannot go to the lab and profile all the potential drug candidates. So that's why our challenge aims at evaluating how useful machine learning is in practice for catalyzing these efforts of compound-protein interaction mapping. So the idea is, or the hypothesis is, that machine learning could prioritize the most potent drug candidates, but also target interactions for further experimental evaluation. So we put the specific emphasis on quantitative modeling of compound-protein interactions, and this is because these are not simple on and off relationships. So we believe that it is very important to fully characterize the activity spectrum of each compound. And furthermore, we focus on protein kinases and kinase inhibitors, and this is because of their clear clinical importance. So kinase inhibitors form the largest class of new drugs that are approved for cancer treatment, but it is also anticipated that lots of other human diseases could be treated using this class of drugs. So actually there already are several kinase inhibitors undergoing clinical trials, for instance, for central nervous system diseases, inflammatory disorders as examples. So the overall aims of this challenge are to determine the best computational approaches for predicting the binding affinities between compounds and kinases, but also the most predictive features for both compounds and kinases, as well as the best bioactivity data for the model training. So all these aspects are important here. So we launched the challenge in October this year, and we have recently completed the first round. But the challenge consists of 2 rounds, and actually the second round is the final one. So we unfortunately expect a delay in launching the second round. So it will be most likely in a couple of months, because the test compounds for the second round are still in the queue for screening in Discovery X. And we will have like a formal announcement about this in Synapse next week. But also I would like to mention that we will not shorten the planned duration of the second round because of that. And we are also brainstorming some potential ideas of like what we could do in between the challenges, having some maybe community phase to keep the participants involved and excited about the challenge. So in each round, the aim is to predict the binding affinities of compound kinase pairs. And specifically, these are dissociation constants, KD values, that we are looking at. So we had 430 pairs in the first round, and we will have 514 pairs in the second round. So this blinded dataset is provided by the IDG-Kinase consortium, and it was selected based on initial full kinome scan screen of 400 multi-targeted compounds, and this was done just as a single compound concentration. So actually the single concentration % inhibition data points are available online, so participants can also try to take use of this data resources. And then what is actually the dissociation constant, KD? It measures how strong is the physical interaction between the 2 molecules, here compound and kinase. and specifically it corresponds to the concentration of a compound at which 50% of kinase molecules exist in a compound kinase complex. So to assemble the training data, we encourage the participants to take advantage of the Drug TargetCommons platform, DTC in short, which is a community effort to curate, annotate, and harmonize the bioactivity data that are currently existing in other databases. So at the moment, DTC contains the bioactivities of almost 2 million compounds, and all this data is publicly available. And also this single concentration % inhibition data points for the test compounds that I mentioned, they are also available in DTC. But of course, participants can freely use any other data resources, for instance, Campbell, PubChem, Pharos, and Drug Target Explorer, to mention a few. We have a baseline model in the challenge, which comes from, which comes from our recent publication. So it is a pairwise kernel regression model where we used only KD values for the model training, and most of these values were extracted from DTC. So in this model, instead of using compound and kinase features directly, we calculated a pairwise kernel based on them. So such kernel can be interpreted as a similarity measure between compound kinase pairs. And to calculate the kernel, we used standard binary molecular fingerprints of compounds and full amino acid sequences of kinases. And we have selected 6 evaluation metrics to score the submissions. So we have root mean square error, Pearson correlation, Spearman correlation, concordance index, F1 score, which is calculated using a single interaction threshold to binarize the KD values, as well as the average area under the curve given several different interaction threshold values. So the submissions in the first round were scored across all the 6 metrics, but they actually remained unranked, so we don't have a winner here. So the idea was to basically let the participants see how well do they perform across all the 6 metrics. And then the final second round will consist of 2 sub-challenges, and the winner of each sub-challenge will be determined based on one of the 6 metrics. So these metrics will be announced definitely before we start the second round, but they have been selected already before we have launched the challenge. So at the moment, we have over 300 registered participants and 45 registered teams. And during the first round, we have received 170 submissions from 77 different competitors, meaning teams or participants who are not registered in any team. And we also have one of our own submission, which is like the baseline model submission. And here is the summary of the results in the form of histograms per each evaluation metric. And you can see that the red lines mark the baseline results. So you can see that the baseline model is quite competitive, but we observe quite many submissions that are better than this baseline already in the first round. So across most metrics, about 10% of the submissions beat the baseline. So it is really nice to see that we have like many good submissions already. And here you can see that the top performers scored high, not across one or a few metrics, but actually across all of them. And I'm not showing RMSE here on this heat map because of the difference in scale, but this observation is also valid there. And then actually we have a single participant that scored highest across 5 out of 6 evaluation metrics. So here each participant is marked with a separate color, and I'm only showing the names of those who agreed that I do so or replied to my email in particular. And also we have performed a bootstrapping by randomly sampling 430 KD values from the prediction file and then calculating the evaluation metrics for those random predictions. And this whole procedure was repeated 1,000 times to generate a distribution of bootstrapped scores per submission. So here we have only the results for the top performers. As well, I'm showing 2 evaluation metrics as examples, so bootstrap Pearson and bootstrap F1 score. And we also have, so the submissions here are arranged by the leaderboard value, and this leaderboard value is marked with the orange circle for each participant. And we have also the bias factor superimposed on these results. So you can see that, for instance, if we would take a cutoff of 3 for bias factor, which is quite standard, Then we observed that we have seven top submissions that significantly differ from the rest in terms of Pearson correlation, and then 2 submissions for the F1 score. So this analysis will be available in signups with some description. So this is just like preliminary, but I think like most likely next week we will have the full analysis available in signups for the participants to have a look. So in the second round, we will also ask the participants to provide a brief summary of the training data as well as the modeling approach when doing a submission. So we haven't done it in the first round to make it easier for the participants, and that's why I cannot really tell you what models are performing best and what were the best training data and all of that, because we simply don't know. But then later we will of course like analyze all of these results, like knowing what are the models and what are the training data. But also we will look at what was the chemical similarity of the test compounds compared to compounds that were used in the training data, as well as the selectivity index of compounds and the number of training data points per each test compound kinase pair. So these are just examples of what we will look into. And in terms of the last point, so in this figure, you can see that S1 corresponds to the easiest prediction scenario, and S4 is the most challenging one. So this basically depends like if the test and training data share common compounds, kinases, or both. And this, of course, depends on the training data, but I believe that the most of the test compound kinase pairs from the challenge, they fall somewhere in the middle. So these are challenging, but maybe not this like most challenging prediction problem when both the compound and kinase are completely new. So I would like to also mention that anyone can still participate in the second round of the challenge because the participation in the first round is not required. And I would like to thank all the challenge organizers, our scientific advisory board members, all the participants of the challenge, and of course all of you for your attention. And I'm happy to take any questions.
So very nicely done. Thank you, Anna. Questions?
This is a comment as well as a question. So for those of you who've been part of the inner workings of putting together a challenge, you might not know it, but most of the time spent on discussions on our bi-weekly or weekly calls, how often they are, is about the scoring metrics. It's a very hard problem to resolve. And in this case, you have 6 scoring metrics, and you've already decided which ones you'll be using to score at the end. Do you want to comment on why you chose to do it this way?
Well, so basically, we didn't want to reveal the metrics from the beginning because it's possible to like basically optimize the model for the particular evaluation metrics, or we have actually seen it in some of the challenges. So we wanted to avoid this as much as possible. But of course, well, there isn't also like a single best evaluation metric, because if there was like a single gold standard, then we would have definitely just selected this one. But yeah, so mostly it was like to like avoid like fitting this like noise basically like for a particular evaluation metric. But we also wanted to be fair, so that's why we have selected them before the start of the challenge. And so like it's not like that we are like trying now to figure it out based on the results from the first round.
Any other questions? Okay, great. Let's thank Anna again.
Thank you.